There are these cool things called hybridomas developed in 1975 by George Kohler and Caesar Milstein. This discovery earned them the Nobel Prize and made it possible to produce large quantities of identical or monoclonal antibodies for the first time. Now, let's look at the two cells involved. The first is an antigen-specific B cell, the other is a myeloma cell. The B cell is a type of white blood cell whose job it is to recognize a foreign substance and produce antibodies to fight them. Here's the catch though, they don't replicate that well in labs. Myeloma cells are cancerous plasma cells that can divide indefinitely in culture, making them useful in research because, well, they're immortal basically. By combining these two, we get something called a hybridoma. So what exactly is the process of making this hybridoma? Well, essentially, we fuse these two cells using something called polyethylene glycol, or PEG. During the fusion, however, we not only produce hybridomas, but we also produce the antigen-specific B cells and the myeloma cells. Now, the antigen-specific B cells will die over time because they don't replicate, but the myeloma cells are more difficult to get rid of. But basically, we can use something called hypoxanthine aminopeptidine thymidine, or HAT. Now, why would this work? Well, a normal mammalian cell can synthesize DNA nucleotides using two different pathways, the de novo pathway and the salvage pathway. During normal pathways, cells will use the de novo, but without it, cells will use the salvage pathway. But this only occurs if hypoxanthine and thymidine are present. The key to this is using myeloma cells that are deficient in something called hypoxanthine guanine phosphoribosyltransferase, or HTPRT, which is required for the salvage pathway. Now, this cell won't replicate because the de novo pathway is blocked by the aminopeptidine from HAT, and the salvage pathway is blocked because there's a deficiency in HTPRT. The hybridomas, however, live because they are not deficient in HTPRT and can use the salvage pathway until aminopeptidine is present anymore. So why do we need this? Well, they actually allow scientists to produce monoclonal antibodies. These are antibodies that are the same, which is super useful for things like diagnosing disease, targeting specific molecules involved in diseases like cancer infections. So, like for instance, Herceptin, an antibody used to treat breast cancer, was actually created using hybridoma technology. So in a world of cells, hybridomas are like the ultimate super team. That's hybridomas in a nutshell. Maybe now you can fuse with your best friend to become the best versions of yourselves.